Now I would like to welcome Dmitri Petrov, who is originally from Russia, but now from Stanford University. And he said he started to study physics, but then thankfully he changed both the country and the field, and became interested in genetics. And he works in evolutionary biology and genetics. And he said that he was, by tradition and by education, he was always a neutralist, but the data forced him to change into adaptionist. And I can prove after discussing over lunch that it's true, and I very much look forward to his talk to show everybody.
Well, thank you very much for the organizers, particularly Virginie, for inviting me. It's been a great conference already. And a lot of things I wanted to mention at the beginning already have been presaged by previous speakers. It's great, so the introduction will be even shorter. So the beginning, I want to start by making the point that Virginie made in her introduction, that the two fields of ecology and evolution have been traditionally separated, and the intellectual justification for the separation is that ecology traditionally studied very rapid processes. So, for example, lynxes and hares cycling on a few generational scale. And you can think of these interactions happening between fixed species. And evolution was seen as something that's happening over a very long period of time. This kind of sets the values of the species, of the lynxes and hares, but it happens for such a long period of time that you can ignore the short-term processes that the ecology studies. And as a result, even though many departments are, and especially in the States, are departments of ecology and evolution, they're really departments in name only. They're really ecologists and evolutionary biologists, and they really do different things on different timescales. And they tend not to talk to each other and value each other's work. So the question then is evolution in fact slow enough to be ignored in the studies of ecology? And if you read Darwin, there are many claims there that evolution is very slow, kind of imperceptibly slow, driven by mutations of very weak effect over a very long period of time. But of course, also the very first chapter of The Origin of Species is about artificial selection, where evolution happens very fast, right? On a scale of a human lifetime at least, but just few number of generations for the species of interest. Now you might think, all right, so this is just a case of human selection that's particularly rapid, and natural selection will be much slower. But again, there are many examples where natural selection was quite fast. So this is an example of the peppered moth, where selection happened on the scale of a decade, just a few generations. Now you can say, again, this is a case of response to anthropogenic changes, to industrial revolution. And other cases like pesticide resistance, which occurs extremely rapidly. But there are also now studies, observational studies, such as by Peter and Rosemary Grant, by David Resnick, and other people in natural systems, where you can just observe evolution happening on the scales of single generations. So this is a case of Darwin's finches. So we have, I think, right now, quite a bit of evidence that evolution can be happening to a substantial degree, large changes occurring on single generational scales, and these might feed into ecological processes. So in my work, I started in population genetics, which traditionally looks at static patterns and tries to make inferences about the forces that must be involved in setting up these patterns of variation within species and between species. And at some point, I decided to shift and take that work by the Grants and others seriously and see, can we actually study population genetics on these ecological timescales? And I've been working on Drosophila melanogaster. I thought Drosophila melanogaster could be a good system for studying this because the ecologically, Drosophila melanogaster is an organism that in temperate areas goes through these patterns of booms and busts every year. So it goes through very large population size increase during the summer, and during the winter it overwinters locally. So adults, females burrow into the ground and survive until the next year by going into reproductive diapause. And just before I even go further, I'm going to say that all of the work I'll be talking about is collaborative work with Paul Schmidt, who's a professor of biology at UPenn, and his work also, the work that kind of changed my outlook on things, and the way I decided is like, this is something to really take seriously. And the work I'll just mention briefly is from his, the paper that I read. It was a paper in 2006, where Paul did a very simple experiment. He went into the orchards near UPenn where he worked and collected flies through the seasons of May, July, September, and in the field and in a local grocery store. And then brought the flies into the lab, established isofemia lines, and a few generations later, put them into conditions where the flies are capable of going to diapause. And diapause, ability to go into diapause is variable, genetically variable in Drosophila gases. Some strains can go into diapause, and some strains cannot. And he found that the ability of these strains, again, in laboratory condition, common garden, a few generations later, varied a lot from May to September, and then it kind of went up and back, going like in cyclical patterns. And then if you go in the grocery stores, it's fairly flat and low. So again, very similar to the case of Culex molestus that lost ability to go to diapause. So in that case, kind of conditions in grocery stores are fairly constant, and so they lost the ability. So that suggested that there is cycling happening on the timescales of just a few generations, so about 10 generations across a single growing season. And so then, together with a postdoc like Alan Berglund, we went to Paul and said, maybe genomics just became available. It's like maybe we can look for evidence of seasonal fluctuations in allele frequencies in these populations. And indeed, we found, so this is one graph from the paper, we found that there's evidence of maybe 1,000 SNPs that change in frequency regularly from year to year. So this is just 3 years. Again, collections from Paul. We just sequenced them, looked for the SNPs that have this pattern, and had evidence that, maybe, again, a few hundred, maybe 1,000 SNPs cycle on all chromosomes, and by substantial degree, so on the order of percents from year to year, from summer to winter. Again, all of this work was spearheaded by Alan Berglund, who is now a professor of biology in Virginia. All right, so the next step was, is this weird thing that's happening just in this one particular orchard, or can we find systematic behavior like this across other populations? And this was work that's spearheaded by former graduate student Heather Machado, who's now working at Sanger. And we'll look at a bunch of populations sampled in the spring and the fall across the United States and Europe. And long story short, I just want to get kind of very quickly go through it. Long story short, we found that there are variants in these populations that systematically go up and down in parallel. This happens so fast that they must be going in parallel across different populations, Vienna and Kansas. And also, kind of first inkling that something may be more going on than just seasonal evolution is that when we did the kind of leave one out analysis, we took all of the populations, took one of them out, identified SNPs that had this behavior, and asked, what is the behavior of the SNPs in one population we left out? And we found that the majority of them, kind of these red ones, if you go to more and more stringent set of SNPs from the population where you define the SNPs, and you look at this leave one out, they have stronger and parallel behavior. So they look to see the SNPs also cycle in the same direction. But there are some that showed strongly anti-parallel behavior. So the SNPs went in the wrong direction. And then we found that you can explain whether population is going to be parallel or anti-parallel by the temperature before the sampling. So it turned out that some of the spring samples were so far into the summer that they started going the other direction. So kind of it looks like there is some sub-seasonal behavior within this population. It's very weak evidence, but there looks like something's going on that's faster than a season. All right, so then, and this is, even though we're working on this very much This is not my idea. It was totally Paul's idea. There's like, okay, we can sequence these populations, until the end of days, but it's very hard to really prove anything in these cases because one problem in particular, you never know when you sample in these orchards whether or not some of the patterns are driven by local migrations, maybe local refugia. You don't understand fully the connectivity between the populations. So it'd be really nice to have more replicated systems. And Paul just decided to bite the bullet and set up these huge cages. There are 2 meters by 2 meters by 2 meters, cages with a tree inside, shows here, with Paul's graduate student Skylar coming out of the cage. So that's the size of the cage. The only food that the flies get there is the food that you bring in, so it's a lab food. And then the flies can live in these cages all through the summer, and Paul keeps them until Christmas. So the flies can maintain themselves until very, very late in the season. And he now has 60 of these cages where the flies can evolve over a single season, and we can watch it happening, and then the parallel setup of the cages allows us to look for the variants that change in their frequencies in parallel, phenotypes that change in parallel, et cetera, et cetera. It's a very controlled system. And so I'll just tell you about one experiment. So in this case, so again, these cages are 2 by 2 by 2. In this experiment, there are 10 independent cages. What you do is you start with a set of inbred lines. So the inbred lines were from natural populations. There are 70 inbred lines, so kind of 70 snapshots of genetic diversity in the population. Before the experiment, you take all these inbred lines, you cross them out with each other for a few generations, 4 or 5 generations, so you end up with this outbred hybrid swarm of these flies, but you know all of the haplotypes because they sequence these inbred lines really, really well. So at the end, you have this hybrid swarm of 70 different haplotypes with recombination happening in before you set and during the evolution. So each cage is started with 1,000 flies from this hybrid swarm, and then you just let them out, let them go. So whatever's happening, day-night variation and variation during the season, they experience all of that. You don't do, in these cases, you don't do any manipulation with them. They just evolve into whatever's happening. And they systematically, so this is a graph of the population size, they all kind of, and this happens year after year, they start at some population size, then they increase in population size, get to some kind of maximum carrying capacity. The carrying capacity of this cage is about 100,000 flies, on that order. Then they persist in that kind of carrying capacity, and then eventually it gets cold and population sizes decline, and eventually they go extinct. So it's this kind of very predictable ecological population size dynamics. All right, so in this particular experiment, what we did, we went once a month, sampled, took the eggs, brought them into the lab, again, raised the flies to the 3rd generation to phenotype them. So this common garden, it's just what's happened kind of in the common garden condition in the lab, and also sequenced a few hundred individuals per time point per cage, figure out their allele frequencies really precisely, and then we looked for the patterns of allele frequency change between for the individual month and across the whole period of time. So both phenotype and genotype. Again, all of this is done with Paul. Done with Paul, and I just lost the names. The work was spearheaded by former postdoc in my lab, Sharon Greenblum, and a lot of Seth Rudman on the right, and Subhaj Japorit, who did a lot of the work with the cages themselves. All right, so what do we find? So the first thing we find is that the phenotypes evolve very rapidly and recurrently on the scale. So for example, fecundity goes from these females laying 20 eggs per day to over 40 eggs a day. So the fecundity increases a lot, and it's on the scale of this experiment. Egg size changes. Developmental rate goes through some non-linear behavior. First it gets much shorter, then it gets a little longer, then it becomes shorter again. There's just a lot of dynamics and exceptionally rapid changes for these key life history phenotypes. We don't fully understand, we don't understand, even not fully, the genetic basis, but these are happening recurrently, and for many of these phenotypes, they happen year after year. So like I said, pigmentation evolves every single year. The flies start dark, get light every single year. Okay, so we know the phenotypes evolve, and then what I was interested in is what's happening at the level of the alleles. So first I'll tell you kind of one analysis that kind of convinced me that there was there. It's a little involved, so bear with me. So what we do in this particular analysis, it's kind of statistics in this time series data is very complicated, so we wanted to simplify things and just make things as robust as possible to any kind of ascertainment biases and any regression to the mean. So what we do is we take a set of cages, we have 10, right? So we take 9 cages, and then we identify all the variants that systematically change in frequency for any particular period of time. We take the SNPs, whatever they are, and then we ask what's happening to these SNPs on average in the left out cage. So the SNPs identified in 9 cages, but they're measured, their behavior is measured in the 10th. So they're entirely independent measurements. And then we compare the behavior of these SNPs that we found with a matched set of controls that had the same starting allele frequency, the same recombination rate, the same. So empirically, how much noise there was due to whatever facts that were happening. So the kind of SNPs that moved in parallel and SNPs that have the same features as the ones that moved in parallel. And so in this particular case, for example, these are the SNPs that were identified during this first period of time, the first month, and then the orange dots is the median shift in the left out cage. And you can see, and if there's a color, it means that it's significant. So in this particular case, every single left out cage, these are all different sets of SNPs, moved in parallel directions. So there's a lot of parallelism during the time that you pick them. But then if you look at other times, sometimes they move in the right direction, sometimes they move in the opposite direction. The crosses are the match controls. So that's kind of roughly the noise. And you can see that sometimes they go in right, kind of during the time you define them, they go in parallel. And other times they go back up and down. So these are SNPs defined during the first month. If you define them at different times, let's say during the second month, you see very similar behavior, 3 to 4 similar behavior, 4 to 5 much weaker signal, and this is 1 to 5 from beginning to end. They turn out to be very distinct set of variants. And you find that quite often you see parallel behavior, but let's say here you find that during 3 to 4 period of time, they're parallel, but during 2 to 3 and 4 to 5, they go in the opposite direction. So these are SNPs that go systematically, let's say, up during this particular month, but they go in a left out cage, they also go up, but before and after they go in the opposite direction. So they are advantageous, let's say, during this month, but they're disadvantageous. They flip their selection coefficient before, the month before and month after. It kind of suggests a very strongly fluctuating selection happening on these time scales, right? So something that could be adaptive one month, it could be maladaptive at other months. Did it make sense? Okay. So, and again, it feels like a very robust analysis to me. So I started believing all of this. And selection that's implied by these median shifts is exceptionally strong. So the SNPs move by percents, but again, this is just 1 month, right? So it's a few generations at most. All right, and then just very quickly, if you then look at the peaks that you find during different months, you find that different, in this color, they're colored here, different windows of time, you end up with completely different peaks. So different alleles change their frequency, in 1 month, then another month, and another month. Sometimes they can happen kind of in the same place for 2 months, but generally it shifts. So different variants are adaptive at different months. So it's extremely dynamic process. Right? So this is what if you look from beginning to end, and this is look individual months. That also tells me that it really matters how you find the variants you're interested in. Because if we only sampled beginning to end, we would have missed all of that behavior. So all of that intermediate behavior is gone because a lot of these variants, even though they're very adaptive to move by a lot during 1 month, they might be going backwards during the other time. And so they average out to nothing. So, but kind of this sampling over a short period of time, you start seeing things. And again, replicated nature of these cages. Okay, so the final thing, so, and this is not published, so everything I talked about before has been published. This is entirely hot off the press and also may not be true fully, right? So this is kind of the analysis that's ongoing. So the paper that we published in this was from the work done in 2014. And then in 2021, we repeated the basic setup, the basic experimental setup. So it's roughly the same strains that went into the cages. It's roughly the same number of cages. So in this case, there were 12 cages rather than 10. I didn't change the graphic. There are 12 cages, so roughly the same number. The only thing is that now we decided, like, we see all that stuff happening on a monthly scale, and it's like, okay, let's bite the bullet and let's sample them weekly, because like every time we look at a shorter window, we see more and more. Is there more happening even on a shorter time scale, if it's even possible? And so we sampled many time points, roughly weekly, until the end where it became biweekly. But you can think of them as weekly samplings, and there are more time points. And this was entirely heroic effort by a postdoc in my lab, Mark Bitter. All right, so the first, just a few slides, I'll show you, we're trying to see whether these patterns replicate from year to year. Again, completely different year, different weather, there was a hurricane in the middle, just different things happening. But at least in some sense, they replicated that the population sizes go up, then they get carrying capacity to go down. So ecologically, it's similar, and maybe similar in some coarse-grained way. It was the summer, and then it got colder. But in details, it's different. So there are kind of a lot of analysis. I'll just show you a few. So first, do we see evidence of evolution happening on single week scale, longer? And do we see evidence of fluctuating selection? So I'll just show you just a little bit of data. So what we do here is that we look at all of the SNPs that we find from beginning to end of the year, right? So beginning of the sampling to the end of the sampling. And again, this is a leave one out analysis. So we find SNPs with 11 cages, and we look at the behavior of the 12 cage, right? So again, so it's entirely independent. So this is what you get if you look 1 to 11, and you measure it. So the SNPs are found 1 to 11, and you measure it in leave one out cage, also in 1 to 11. You see a lot of parallelism. So the dots are the true sites, and the crosses are match controls. So there's a lot of parallelism. Then we start getting progressively shorter windows. So instead of 1 to 11, 1 to 10. And we see that it's still a lot of parallelism, but it gets weaker. So the effect, the movement gets smaller, as it should, because they move by a certain amount from 1 to 11. You look at a shorter window, the total displacement should get shorter. So it goes like this, 1 to 10, 1 to 9, 1 to 10. So the total displacement gets shorter. It's still significant. And then we'll start getting to single week resolution. And in single week resolution, you start seeing both parallel and anti-parallel behavior. So the crosses again are match controls, but you see shifts, systematic shifts on a single week scale. Again, SNPs are found beginning to end in 11 cages and measured in the 12 cage in single weeks. And you can see that sometimes they move in the positive direction, but often they move significantly in the opposite direction. So there's fluctuating selection happening in individual weeks. I'm not going to show you the data, but when we look carefully, some of the SNPs change the sign of selection coefficient like 7-8 times during the summer. So they're adaptive, maladaptive. It's extremely dynamic. All right, so now we're going to look at parallelism. So what we're doing here is we say, we're going to take all the SNPs we found in 2021, and we're going to say, are they in any way predicting behavior in 2014? Are they going in the same direction or in the opposite direction? And we're doing permutation scheme to make sure that kind of all of the noise is controlled for, but we shift the identity of the SNPs. And the gray bar is what we get during permutation. And what we're measuring here is the correlation coefficient in terms of the movement of the SNPs in 14 and 21. And we find that when we look at the true sites, they often are very substantially correlated, and often they're very substantially anti-correlated. But when you pick SNPs in one year, it says this SNP's going to move a lot in the other year. You just don't know in which direction. They sometimes move in the right direction, sometimes in the opposite direction, because the selection is so fluctuating. And you can go further. You'll say, okay, We have peaks in both years, and we're not sure how many of them are false positives, false negatives, we don't know. But we have about 160 genes identified in 1 year, and identified, you find the peak and you find the central point on the peak and say, okay, let's just guess that's the gene. I've no idea whether it's true or not, but that's my best guess. And I have 165 in 1 year and 1,000 another year. And I ask, what is the overlap between them? And by chance, I should see about 10 genes overlapping, and in fact, we see about 40. And a lot of these peaks are so wide, I don't believe them. So the fact that there's any overlap between the genes, so like roughly 30 of these genes turn out to be probably real overlap. So it's like 30 out of 165. So it's extremely parallel evolution at the level of genes, at least, at the level of SNPs, and the level of genes in 2 distinct years. We can then look at what are the functional categories in the 2 years. And if we looked at 2014, we take this 165 genes and we put them through a database asking, is there any enrichment for any function? And the key enrichment that shows up for us, the only enrichment that shows up, is enrichment in genes that involve in neuronal development, neuronal function, and synapse formation, kind of neuronal behavior, and so just a bunch of categories. And in 2021, we see the same thing. In the overlapping genes and in non-overlapping genes. So it looks like in both cases it picks the same functional sets of genes. The last thing I'll say, that was, I really didn't think it was going to work, but we just took the SNPs and we found SNPs that are adaptive, according to our standard, at different phases during 2014. So, early, middle, late. And we ask, if I take the SNPs and I look for which period of time is best correlated from here to here, and do I find any matching? Do I find, let's say, early exponential matching early exponential, carrying capacity matching carrying capacity? It's not fully clean, but it's pretty clean. So the first month predicts the beginning 2021, and in general, they go in time staggered way. So the SNPs themselves predict the period of time ecologically. I don't know whether in terms of temperature, I think this is first analysis, but ecologically you can predict what is going to be happening. So just to conclude, I think our data is not the only data. There's just no way to separate the two time scales. Evolution is happening on such a rapid time scale, and it changes phenotypes by so much that you cannot ignore evolution happening on the time scale of a single boom and bust cycle, of a single exponential to carrying capacity. So that's just not true, and we should stop doing this. And I think that what's happening in all of the system, we find that if we look at a very short time scale, we find selection is extremely strong. It's directional selection. However, it's inconsistent. It goes back and forth. The strength of selection is always high, but the direction is inconsistent. So at any given time, you have very rapid evolution, but long term, you have stasis. This Drosophila monogaster doesn't evolve to a new species over 3 seasons. They just basically stay put. Again, just to say, there's a long-term collaboration with Paul. It's been great, and hopefully it will continue for many years into the future. And just want to thank my lab, and thank you for listening.